Hello everyone and welcome to Instant Biology by Dr. Neelab. I am here with you again with another very important topic that has been requested by many students. So the topic that I would be dealing today is transfection, and to be more precise I would be dealing with transfection through calcium phosphate precipitation method. So a lot of students find difficulty in the process in understanding the process of transfection. So what I'll be doing is I will be firstly explaining to you what transfection is, and then I would be telling you what are the different methods through which one can do transfection, and after that I would be focusing specifically on the process of transfection through calcium phosphate precipitation method. So without much ado, let us start. Just before starting, I would inform you that I have already made a video on this method in Hindi. That was long back, but I'm just making this video for students who understand English better. So this video would be in English. If you understand Hindi better, I would be placing the link of the video on the top at the last of this particular video. So let us start. So what is transfection? Let us first of all understand the process of transfection, or what transfection is all about. So transfection is the process in which we introduce DNA into animal cells or embryos. That means a foreign DNA, let me just write it down over here. So what happens is a foreign DNA, it is introduced inside a animal cell. Now this is an important point. When the foreign DNA is introduced inside animal cell, or you can also mention embryo over here. So it is similar to the process of transformation. It is a process that is similar to transformation, but transformation term is specifically restricted or reserved when you are transferring a particular DNA fragment inside a prokaryotic cell. So in other words, when we are transferring a DNA fragment into a prokaryotic cell, then the process is referred to as transformation. But when we are doing the same kind of a thing in case of an animal cell or eukaryotic cell, then the process is referred to as transfection. Okay, so this is the basic difference. Apart from that, the gene transfer methods can be classified into multiple types. For example, if I'm talking about prokaryotic cell, you can transfer the genes through multiple methods. The first method was conjugation. I haven't talked about conjugation as yet in English. I've already done that in Hindi, so you can check out, if you want to study that, you can check out that video. I would be making videos in English also now: conjugation, transformation and transduction. So next method is transformation, and the last method is transduction. So these are the three methods through which you can transfer the gene fragment, transfer a DNA fragment inside a prokaryotic cell. We would be talking about these in the upcoming lectures, but today, since this lecture was requested by a lot of students, so I have decided to take up this lecture today in today's class. So what is now coming back to, since I have introduced the topic of transfection, so it is the transfer of DNA fragment into animal cells or embryo. Now this can be done through various methods. Firstly, why would you want to transfer your DNA fragment inside an animal cell or inside an embryo? The chief requirement or the chief advantage of doing so is you want to create a genetically modified organism. You want to create a transgenic organism. You want to create an organism that would be beneficial for your use, for the mankind. You might be knowing that different plants are being modified, different animals are being modified for different purposes, for the benefit of mankind. Now this is the prime vision or prime requirement why this is done. So this is the reason why transfection actually is performed. Now, what are the different ways or methods through which transfection is performed? Now, there are different methods, as I have already said. One is calcium phosphate precipitation, that is the most commonly used methods in lab. Next is fusion with bacterial protoplast, microinjection, retrovirus infection, lipofection, particle gun delivery, and electroporation. So these are the different methods. In today's lecture, I would be restricting my discussion to the first method, that is calcium phosphate precipitation method. Now, let us come to the topic that I want to discuss today, calcium phosphate precipitation method. I would be showing you certain diagrams also. I would be showing you or giving you the flowchart also, how this is done. So, how is calcium phosphate precipitation method performed? Now, what you do is you prepare the DNA. That means you isolate the DNA, you prepare the DNA that you want to use in the process of transfection. Now, of course, you will have to isolate the DNA from this source organism or from the donor organism. Now, suppose you want to take out the DNA from a human being, that is the source of the DNA. You will have to firstly isolate the cells from the human beings. Let us say you want to isolate it from the WBCs. You take out the WBCs, you rupture the WBCs, and then you take out the DNA, you cut it through restriction endonucleases and find out the exact fragment that you want to isolate. Then this fragment is actually called as the DNA preparation, because this is the DNA fragment that we want to transfer inside the animal cell or embryo, for the fact of the matter. Now, this DNA preparation is added to the phosphate buffer. Now, firstly, this is done. That means the DNA preparation is now put inside the phosphate buffer. Now, phosphate buffer is a very commonly used buffer in the labs. So that is what you do. You add DNA material or DNA preparation to the phosphate buffer. After that, this is the first thing that you do. You mix these two, and then this and this is mixed next. That means firstly you add DNA preparation to the phosphate buffer, and after that what you do is you add calcium chloride to that. I would be giving you the explanation behind why it is done in that way in just a moment. Firstly, let us go through the flowchart. Now, then what happens is insoluble calcium phosphate will be produced. Why insoluble calcium phosphate will be produced? Because you see, calcium chloride CaCl2, it will be breaking down into Ca2 plus 2Cl minus. So this calcium ions would be attaching to the phosphate. So this calcium ion would be attaching to the phosphate, and this would be forming a insoluble calcium phosphate. Insoluble. Why it will be insoluble? Because its density would be higher than the surrounding liquid. So what will happen? It will try and co-precipitate with the DNA. It co-precipitates with the DNA. What do you mean by co-precipitating? That means along with the DNA it will be precipitating. That means DNA as well as the calcium phosphate, insoluble calcium phosphate, they will be precipitating together. This is called as co-precipitation of the DNA. We will be understanding this in much more detail as we move forward. Next process is then this entire material or co-precipitated DNA and calcium phosphate is added to the cells. What do the cells do? Cells do the phagocytosis. Now, what do you mean by phagocytosis? They will be engulfing the calcium phosphate and DNA complex. They will be engulfing it. Something like this would occur. So suppose this is a cell. This is a cell, and let me just show this. So suppose this is the calcium phosphate co-precipitate along with DNA. So what will happen, because this calcium phosphate DNA complex would be attaching or would be present on the surface of the cell. So what will the cell do? It will form a something like this would be formed, and this co-precipitation would be engulfed like this. It would be engulfed, and when it is engulfed, it is taken inside, and the DNA is freed, and DNA can be used up by the cell. We would be talking more about that. So this is, I mean, the DNA is taken in through the process of phagocytosis. This is an important point. You should be knowing about this. Now understand this, roughly 20% of the cells can undergo transfection through this approach. Now this is the 20% that we are mentioning over here, that has been obtained by a lot of modifications on the protocol. Initially, when transfection came into being, only 1 to 2% of the cells were able to get transfected. But later on, as the protocol got modified, more than around 20% of the cells started getting transfected. So this is an important thing. Next is small proportion of the cells stable or permanent. I mean to say is that a small proportion of the cells, now suppose you are taking 100 cells in original, a small proportion out of these cells will undergo stable or permanent transfection. Now what do you understand from permanent transfection? In permanent transfection, what will happen? The DNA will be attached to genome of the cell. What do I want to say? The DNA that you have transfected inside this cell, that particular DNA would be attached to the cell's genetic material. This is referred to as stable or permanent transfection. One more important thing needs to be kept in mind when you are doing transfection. This is aeration of phosphate buffer is important. That means you continuously provide air inside the phosphate buffer. Why? Why is that so? Because when you are adding DNA to phosphate and then you are adding calcium chloride, what happens is there are chances that clumping might occur. Clumping of DNA calcium phosphate might occur. Now if the DNA calcium phosphate clumping takes place, that means a lot of DNA fragments are clumped together, then the probability or possibility of engulfment of this complex becomes very less. So you would always want the transfection chances to be improved or to be very high. So you would not want this clumping to occur. That is why continuous aeration is provided. I hope this concept is clear to you. Now let us move on to the advantages, or rather let us move on to the diagrammatic representations first, and then we would be talking about the advantages and disadvantages. Now you can see that this diagrammatic representation clearly depicts that what is happening. So in the first image you can see that this is the DNA over here. This is the DNA. Okay. Now this DNA, because then what you are doing is you are adding DNA to the phosphate buffer, and then you are also adding calcium to it. So first of all, what happens is, because DNA is negatively charged, you can see that this DNA is negatively charged. Why is the DNA negatively charged? Because of the presence of the phosphate ions, right? Now what happens is, because of the negative charge of the DNA, calcium gets attracted over here and calcium starts binding to the DNA like this. This makes the DNA very heavy. Now also the phosphate groups also now start getting attached over here. Why? Because calcium ion is present, calcium is having the positive charge, and phosphate in the medium is also present over there. Though phosphate will also get attached to the calcium ions over here. So addition, and this what will happen over here? The DNA, this is the DNA, along with the DNA, calcium phosphate will be added. So these blue colored balls over here are showing calcium phosphate that is attached to the DNA. Now because of the presence of the calcium phosphate attached to the DNA becomes very heavy, and because of this the DNA calcium phosphate complex will be settling down, or it will be precipitated. This is the reason. I hope you got it clear. Another diagram, this is the diagram from B.D. Singh. You can check out that this is a cell, and the cell is added to DNA, the cell is added to the phosphate buffer, and then firstly the DNA is present inside the phosphate buffer. Let us understand in this way: the DNA is present inside the phosphate buffer, and you add calcium chloride onto it, as I said in the earlier when I was describing the entire process. Because of this, what will happen? The DNA was interacting with the phosphate, and now calcium will also enter. So DNA calcium phosphate co-precipitation will be occurring. Now you will be adding this entire thing to the cell, and now you can see that this is the DNA calcium phosphate precipitate, and now this is entering inside the cell through the process of phagocytosis, and then transgene construct integrates inside the genome of the cell. That means the original DNA of the cell, the gene that you have entered, or the DNA that you have transfected, will go and integrate inside the genome of the cell. This is what is being said over here. So the diagram is basically representing almost the same thing. Now this is another method, and this method I have obtained, or this diagrammatic representation I have obtained from the Thermo website. So you can check out this chemical transfection methods. In this I am focusing upon the calcium phosphate method first. Apart from this, there are many other methods, but I am currently focusing upon the calcium phosphate method. In this also you can see that the DNA is present and calcium ions are attached to the DNA because of the charges, and then you can see that endosome is created. What is endosome? Because of the phagocytosis, the endosome will be created, and through the endosome what will happen? The DNA will be moving inside the cell, and after that the endosome will be rupturing and the DNA will be separated, and this DNA can be attached to the genetic material of the cell or genome of the cell. So this is the basic method through which the transfection will be taking place in calcium phosphate. Now let us talk about the various advantages of this method. Can be used for transfection of virtually all mammalian cells. Now I have already said that it is a very common method. It can be used for all the mammalian cells. Next, large number of cells can be treated with little or no effort. Now you see that you can address, or you can work on multiple cells at a single time, and you can transfect all of them with little effort, because calcium phosphate treatment is not a very multi-step process, or it is a simple process. Additionally, the components that we are using over here, calcium chloride, phosphate, calcium phosphate, they are very inexpensive, and they can be used, or they are generally used by all the labs. So the technique is extremely simple and inexpensive. This is again an advantageous feature of the entire cell. Now let us talk about the next thing: transient and stable transfection. Now let us understand this transient and stable transfection. What do you understand from this? Transient, that means temporary transfection can also be achieved, and stable transfection can also be achieved. Both of these things can be achieved through this process. So I told you a small population of the cells will achieve stable transfection, and some population will also achieve transient transfection. Now let us talk about the disadvantages of the entire process. Variation is possible. Now understand this, that this calcium phosphate precipitation method is actually dependent upon some parameters such as temperature, pH, buffer concentration, etc. Therefore, if only a small variation is there, such as temperature, pH, buffer, it can lead to variations in the result that you are achieving of the transfection efficiency. That means with a little variation in these parameters, the transfection frequency can vary. So variation is possible. Cytotoxic to certain cells. Understand this, that certain cell lines do not appreciate the use of calcium phosphate, because the calcium phosphate proves to be cytotoxic to these cell lines. So you cannot use this method for them. Unsuitable for in vivo transfer. What do you mean by in vivo? In vivo simply means directly inside the cell. You will have to bring the cells out from the body of the organism. You cannot directly put or you cannot directly transfect the cells in under in vivo conditions. You'll have to isolate the cells and then do the transfection, and then you can place the cells back inside the body of the organism. Relatively poor transfection efficiency. I have written relatively over here. Relatively simply means that compared to the other chemical transfection methods, the transfection frequency is not that high. I have already told you approximately 20% transfection occurs. So these are some of the advantages and disadvantages of transfection method. So I believe you would have got a clear understanding of calcium phosphate precipitation method. The next class I would be coming up with another method. So if you like the lecture, please hit the like button. Do not forget to subscribe, and also tell us which topics you would want to study next. Thank you so much and have a good day.